So I'm going to talk about some topics that have been in the news in the last couple of years quite a bit. And by way of introduction, let me say that the human story cannot be told without recounting the suffering caused by infectious diseases. Viruses in particular have especially wreaked havoc, particularly in the post-agrarian world. So when our ancestors learned how to grow crops, they began to live in larger communities. And so viruses that could be spread by casual contact could be transmitted to many others. Furthermore, in these large communities, new births and also interactions with other communities that they traded with provided a constant supply of susceptible people. So viruses could not get easily extinguished by either killing off the entire population or making everybody immune, as happened when we lived in much smaller groups as hunter-gatherers. We also started to domesticate animals, and so the pool of viruses that used to circulate in animals also started to circulate in humans. And thus it came to be that diverse viruses that can spread by casual contact and which can be reasonably deadly, such as SARS-CoV-2, began to circulate in humans. Urbanization, population growth, the Industrial Revolution, and travel exacerbated all these effects. So here we are today. How did we survive as a species? We survived because we have an immune system, and we have an adaptive immune system, which is remarkable in that it enables us to mount pathogen-specific responses against a diverse and evolving world of microbes. Isn't it amazing that if you are infected with SARS-CoV-2, you can mount a tailored response to this virus, which evolved after you were born? So to roughly see how this works, consider infection with a virus. When a virus infects a host cell, it hijacks its transcriptional machinery, enabling the synthesis of new viral proteins and the assembly of new virus particles that can go on to infect other cells. Now, B cells are important players in adaptive immunity. We have about 100 billion B cells, and most B cells have a receptor on their surface that is distinct from the receptor on the surface of another B cell. So if the receptor on this particular B cell can bind sufficiently strongly to the spike proteins of this particular virus, biochemical reactions cause it to get activated, that is, new gene transcription programs turn on, and then it starts to replicate, and a Darwinian evolutionary process takes place, which I shall describe a little bit later, which ultimately leads to secretion of soluble and even higher affinity forms of this receptor, and that is what we call an antibody. These antibodies can then bind specifically and strongly to the spike proteins of this particular virus and then dispose of it in a number of different ways. But antibodies principally do battle with free virus particles in blood or extracellular spaces. Now, some of these viral proteins that are made inside the infected cell get chopped up into short peptides, about 10 amino acids long, and they are displayed on the surface of these infected cells in complex with human proteins called HLA proteins. T cells are also important players in adaptive immunity. Again, we have about 100 billion T cells, most of whom have a receptor that's distinct from that of another T cell. And if the receptor on this particular T cell can bind sufficiently strongly to this viral peptide, it gets activated, it starts to proliferate, and then can coordinate an immune response in a number of different ways. For example, certain kinds of T cells called CTLs are such that if an activated CTL sees another cell displaying the same peptide that originally activated it, it can secrete products that will kill this infected cell and the virus particles that it harbors. Now, after you have cleared an infection, most of the B cells and T cells that multiplied in response to this particular infection die. But a few remain as so-called memory cells, which can mount robust and rapid responses upon reinfection with the same pathogen. It is this pathogen-specific immunological memory that is the basis for vaccination. With a vaccine, you try to induce memory B cells, T cells, and antibodies that are specific for the bug against which you wish to protect the host. Now, vaccination has saved more lives than any other medical procedure. And indeed, today, the reason we see light at the end of this pandemic tunnel is partly, if not largely, because of the availability of effective vaccines. But we don't have effective vaccines against many highly mutable pathogens. HIV is a prominent example. We don't have a universal vaccine for the seasonal flu either. So every year we have to make a new vaccine to combat new strains that evolve every year. We have already seen the emergence of variants of concern for SARS-CoV-2. If it ever becomes true that global vaccination efforts succeed, then there will be enormous selection pressure on this virus to evolve mutations that evade these natural and vaccine-induced immune responses. So the way to eliminate such scourges from this planet are really universal or variant-proof vaccines. And the work that I'll briefly describe today represents small steps in pursuit of this quest. So a major barrier to the quest for a vaccine against HIV, for example, is the high mutability of this virus. In these phylogenetic trees, the number of Ns obviously corresponds to the diversity of circulating strains. And you can see that the diversity of the flu virus in the entire world in a particular year is comparable to the diversity of HIV strains in a single infected individual, and completely dwarfed by the diversity of strains in just one region in Africa. So this high mutability presents a challenge for vaccination because HIV can mutate to evade vaccine-induced antibodies and T cells that lie ready and waiting for just a few strains. So today, my focus will be on the antibody arm of this challenge, which I shall describe in terms of some general principles, and then illustrate it briefly at the end in the context of influenza. But I'm going to spend a slide or two to provide a teaser on the T-cell arm of this challenge in the context of HIV, which we have worked on for over a decade. And the purpose of this teaser is that to motivate some of you, if not, or maybe no one will be interested, in asking questions wherein the issues really pertain to the focus of the last session yesterday afternoon. So because of the high mutability of HIV, there has long been an interest in focusing a vaccine-induced T-cell response to target peptides containing sites which exhibit low amino acid variability when you line up sequences of the same HIV protein derived from virus samples extracted from diverse patients. The idea, of course, is that if a T-cell targets a peptide containing such a residue with low amino acid variability, then HIV would have to evolve a mutation there to evade the T-cell pressure, but that should likely make the virus replicatively unfit, because why else do you see low amino acid variability at that residue? But the trouble is that this strategy can be blunted because due to its high mutability and due to its very high replication rate, HIV can often find other mutations, so-called compensatory mutations, that can partially restore the fitness cost incurred by making the primary immune-evading mutation. So now you have a virus that's almost as fit as before and has evaded the immune response. To define the mutational vulnerabilities of a virus like HIV, you have to determine these collective compensatory pathways that the virus uses to evade human immunity in order to not target them with a vaccine-induced immune response. And you also want to determine the combinations of mutations that the virus cannot make and remain viable in order to target them with a vaccine-induced immune response. So to address this challenge, over a long period of time, we tried to translate the data on thousands of sequences of HIV proteins into knowledge of their fitness landscape. That is, the knowledge about the ability of the virus to replicate and propagate infection in humans as a function of sequence, but with explicit account for the effect of coupling between mutations. We did this in two steps. As a first step, we developed what I call a prevalence landscape. That is, we translated the data on thousands of sequences using a maximum entropy-based learning algorithm into knowledge of the probability of seeing a sequence Z in circulation. So that's sort of like machine learning, if you like. But the prevalence of a strain is influenced by its intrinsic fitness and how human immunity at the population level has pushed the virus population around. So we have to deconvolute these effects because we seek the intrinsic fitness landscape, because we then want to push and pull on it with a vaccine-induced T-cell response to corner the virus between either getting killed by the T-cell response or evolving suicidal mutations. So there we developed a mechanistic model of the evolutionary history of HIV, and we deconvoluted these effects, and then the intrinsic fitness landscape thus obtained was tested with collaborators against in vitro experiments as well as clinical data. And encouraged by these comparisons, we designed an immunogen, the active component of the vaccine, to be made up not of whole proteins as is traditional, but made up of only pieces of the proteome, which are what we call multidimensionally conserved. That is, it is difficult to evolve mutations there without a large fitness cost in all possible sequence backgrounds. Now, you take these bits, if you deliver them into an organism, such as a human, will they mount a strong immune response to these bits? Many people don't mount strong immune responses to these bits. So, in collaboration again with other people, we showed that using the vector of the COVID-19 Johnson & Johnson COVID-19 vaccine, in monkeys at least, it mounts strong immune responses against these vulnerable regions. And we are now working with a company to try to move this concept forward to humans using the more flexible mRNA platform. So with that, I'll now turn to antibodies. Upon natural infection or vaccination, antibodies are produced by a Darwinian evolutionary process called affinity maturation. I mentioned earlier that if a so-called naive B cell, its receptor can bind sufficiently strongly to the spike proteins of a virus or any antigen, then it gets activated. It can then seed structures in your lymph node that are called germinal centers. And herein, these B cells start to multiply, and then because as part of activation, a gene called AID gets expressed, it introduces mutations into the antigen binding region of the receptor at a very high rate. These mutated B cells then leave this area, which is called the dark zone, and transit to another area called the light zone, and there they interact with the antigen, which is displayed on other kinds of cells. Now, if the receptor on the surface of what these mutated B cells can bind sufficiently strongly to this antigen, it pulls on it and can eat it up. And then it chops it into short peptides and displays itself on the surface in complex with these HLA molecules. And the B cells that have eaten antigen and display peptide compete with each other to interact with another kind of T cell called helper T cells, and a productive interaction leads to a survival signal. Otherwise, the B cell dies. So you can see that stochastically, a B cell with a receptor that binds more strongly to the antigen is more likely to eat more antigen, is stochastically more likely to display more peptide, and therefore more likely to beat its peers in the competition. Now, a small fraction of the positively selected B cells are let out as either memory cells or plasma cells which secrete this receptor in soluble form, and that's what an antibody is. But the vast majority of the positively selected B cells are recycled for further rounds of mutation and selection. So you can see that as time ensues after infection or vaccination, the affinity of the antibodies for the antigen will keep rising. Now here is a schematic depiction of the spike of HIV. It has some relatively conserved regions, such as that shown in yellow, because they have to bind to human receptors in order to propagate infection, and we are not mutating on this time scale. However, the size of this region is smaller than the footprint of the binding of an antibody, typically, and it's surrounded by these highly variable regions shown in red and protected by these sugars shown in blue. This is influenza spike. It also has a receptor binding domain here that is relatively conserved, but it also has a conserved bit in the stem of this spike, which is important for mediating conformational changes that are important for viral entry. Now, immunologists have been able to isolate from some infected patients antibodies that at least in vitro can neutralize diverse strains of these viruses. these so-called broadly neutralizing antibodies, or BNAbs, bind to regions that contain these conserved parts. The trouble is that these BNAbs usually emerge several years after infection in small numbers. Immunologists call that titers for the case of HIV, and rarely upon influenza infection. That is, the immunodominant response, if you like, is focused on all the variable bits. However, they're existent. The existence of BNAbs is proof of concept that the human immune system can evolve such antibodies, and thus raises the tantalizing possibility that you can induce such BNAbs efficiently in diverse people by properly designed vaccination protocols. Now, immunologists would frame this challenge thusly. Look, the immunodominant response hits something that I don't want. The subdominant response hits something I want. And they would say, I want to change this immunodominance hierarchy, and in order to do that, I'm going to immunize with variant antigens, which share the conserved bits, but the variable parts are different, and hope that this Darwinian evolutionary process will learn how to focus on the common bits. Now, you can equivalently frame this problem in a more physics approach by saying affinity maturation is a highly non-equilibrium dynamical process. The evolutionary trajectories that lead to BNAbs are very rare. I want to immunize in such a way as to provide the right selection forces or external fields on this such that these rare evolutionary trajectories become more likely. And that's the perspective that I will take. This work has been done with a number of former students and postdocs over the years who all have independent faculty positions or are even more gainfully employed. And some students and post-students who are still working with me are here, and has involved collaborations with a number of immunologists as well as physical scientists. And I really want to highlight my many, many collaborations over 2 decades with my colleague, Mehran Kardar. So the computational models that we built to study this problem were in various flavors. One flavor is just agent-based stochastic calculations, which is just implementation of the rules of affinity maturation that I just told you stochastically. But of course, this agent-based computer simulation is just solving the underlying master equations. So we also directly solve master equations or Fokker-Planck equations in some simplistic description of the same processes. All of these things ultimately gives you the probability of obtaining antibodies of different breadth of coverage of a panel of antigens, or the number of BNAbs, if you will. So let me just say, for these agent-based computer simulations, we do not use atomistically detailed descriptions of the binding free energy of a B-cell receptor with an antigen. Rather, we use coarse-grained approaches, and here's one example. So immunologists call the region of the antigen to which the BCR binds the epitope. And so we have a conserved part that is surrounded by variable regions. The conserved regions, each site is described by a spin variable, SI equals +1. And for the variable parts, it can be SI +1 or -1, and thus you get variation. The B-cell receptor sites that interact with them are described by fields HI, and these HIs couple with the spin variable to give you the free energy. This is with the variable parts. That's why SI can be +or -1, and here SI is always 1 because the conserved part. And then we add bells and whistles to make sure some aspects of the conformation of these things is accounted for. These H fields evolve by mutation during this process. Okay? In any case, our work in this area really started with this paper some years ago, where we showed through computational modeling that if you give a cocktail of variant antigens simultaneously, let's say you just make these and put them at the same time, the affinity maturation encounters all 3 of them, then that is a much less efficient way of generating BNAbs than if you immunize sequentially. That is, you give the first one, let affinity maturation happen, you take the memory cells that come after that first immunization, and you give the second shot with the second variant, and then the third shot with the third variant, and so forth. And this, and we describe the mechanisms for it, which have all been tested over the years in experiments running from mice to monkeys. So the question I'm going to focus on now, okay, so sequential immunization is better. Can sequential immunization be optimized, for example, by optimizing the distance, the mutational distance between the variable parts of the antigens you give sequentially. Okay? So that's the question. So when we do these agent-based calculations, we always find there is optimal mutational distance between the variable parts of the immunogens, the antigens, in sequential shots. First or second shot, second or third shot. There's always a mutational distance that optimizes the production of bNAbs. This is one example shown here. This is the mutational distance between the first and second shots. Number of bNAbs is maximized at an intermediate mutational distance. So let's try to understand this result in terms of evolutionary biology and learning theory. So the affinity of a B cell for an antigen depends on many features. The sequence of the B cell receptor, the conformation, as caricatured in the simple model that I described to you. Let us imagine, for making a toy model to understand this result, that there exists some one-dimensional projection of that higher dimensional space that reflects the breadth of each B cell. And that is depicted here. High breadth, highest breadth is here, and as you go towards the edges, there's low breadth. Now here is the viral spike of HIV again. Notice that it has only a few regions that have conserved sites, and most of the regions don't have conserved sites. So if you just directly immunize people with this thing, then there are many more B cells you have that can target all these other regions compared to this one. So just entropically, you're not going to get bNAbs by this evolutionary process. Okay? So you can't do that. So what immunologists do is they've developed very simple antigens, so-called germline or naive targeting antigens, which are very simple, a simple scaffold that contains these conserved regions. So it activates, in the first shot, those B cells that can bind to that conserved region. Okay? These are not bNAbs, but they have the potential of becoming bNAbs. So if you draw the probability of B cells after this so-called germline or naive targeting antigen is given, the probability of B cells of different breadth, they will mostly populate these low breadth regions. But they have a chance of becoming bNAbs later. So now you're going to immunize with these variants. And the first variant will obviously be different from the germline antigen, and the second and third ones will be different because I'm going to put different variable parts. Okay? And that's the process we are going to study. So when you immunize with this full spike, you impose a fitness landscape in response to which this B cell population evolves. Now the fitness landscape can be viewed as the relative probability that B cells of different breadth can replicate. So you can think of this as a probability distribution. Now, this fitness landscape will be peaked for the highest breadth because the second antigen is different from the germline targeting antigen. So the most cross-reactive ones will still be the best. Okay? Now, the B cell population will adapt in response to this fitness landscape. And perfect adaptation would mean that at the end, this probability distribution would mirror that probability distribution. So in a sense, the distance between these two probability distributions is the difficulty of the lesson you have to learn by evolution and how far you have to go to perfectly adapt. And this can be quantified by the Kullback-Leibler divergence between these two probability distributions. Now notice that if the mutational distance between antigens in successive shots is small, then the fitness landscape is pretty flat. That's because the antigens are almost the same, so low breadth B cells that are not very cross-reactive can multiply. So the KL divergence will be quite small. On the other hand, if the mutational distance is very large, then only the highly cross-reactive ones can multiply, because they're very different antigens. And so the fitness landscape will be much more peaked, and the mutation and the Kullback-Leibler divergence will be high. So one way of thinking about this is the KL divergence in this problem, as I've defined it, is also the non-equilibrium driving force that is pushing adaptation. So we're going to study it as a function of D. So for that, one can make a very simple discretized model, where you would take that breadth bin and partition it into some discrete number of bins. B cells can transition from one bin to another by mutation. But some of these mutations can be lethal. And also, the basic state of a B cell is death. So there is also this transition probability from all states to the absorbing boundary condition called death. Now, one important thing, there are many B cells that are of low breadth compared to those that are of high breadth, because cross-reactive B cells will be few compared to all the ones that are strain-specific. So the mutation probability going towards low breadth is higher than the mutation probability going toward high breadth. So there's a constant mutational flux towards low breadth states. So now you can just solve this by writing down the master equations for this problem in a very standard way. And you solve it with the initial condition of this B cell distribution. And you end under one of two conditions. Remember I told you that every time the B cell multiplies, it eats antigen? So at some point, if all the B cells multiply very quickly, they'll eat up all the antigen and this process will stop. And the proxy for that is when B cells exceed a threshold, this thing comes to an end. The other is if there's extinction. B cells can die in any evolutionary process. So if you solve this master equation, which is not hard to do, you will find exactly again, in the function of this D, you will see this optimum. But now it's easy to understand the origin of this optimum. See, when D is small, there is a very small non-equilibrium driving force to become BNAbs. And that is because the antigens are very similar, so low-breadth B cells can multiply. Now, there is this flux I told you from high breadth to low breadth, but they come to the low breadth states and also multiply. So very quickly, the process eats up all the antigen, you stop. If you like, if you think of the B cells as being students in a class, then you know the students pass, but they don't learn very much. Now, if you give a very high mutational distance, very large T, then you see in this region, there is a very large non-equilibrium driving force to adapt. And typically, when there's a large non-equilibrium driving force, you also get a large flux. And indeed, you do in this case. There are a few lucky B cells that quickly make these adaptive mutations that gets them to high breadth, and they survive. But they are a minority. And they're a minority because the antigens are very different, so these low-breadth B cells don't really multiply. And when this flux comes from high-breadth states to low-breadth states, they meet this absorbing boundary condition called death. And the typical answer here is all the B cells die. Okay? When you're right at the optimum, what is going on is that these B cells with relatively low breadth, I can make this quantitative in discussion if you like, they can multiply with some reasonable probability. And when they do, it's with some probability they make mutations forward. And so this flux going forward and the flux going backwards are better balanced. So there aren't extensive failures. You don't die immediately. But neither do you quickly eat up all the antigen because you're not succeeding much. And so you can slowly acquire all the many mutations required to get this cross-reactivity. So the B cells pass and they gain knowledge, if you like. Now, there's another thing that I show you. This is now, for various values of D between the germline targeting and the first shot, I have graphed here the calculation results for BNAb titer numbers as a function of the mutational distance, or D, between the first and second shots, if I give a second shot. You see that, again, there's a peak, and if this turns out to be the optimum value for the first shot, for that value, you see there's another peak for the boost. So there's an optimum prime boost protocol, if you like. Okay? It turns out, and again, if there's interest, I can describe it, this optimum is really set by what happened during the prime. That really sets what kind of situation you have so that you can expand all these B cells. But the point I want to only emphasize here is that notice the scale compared to that scale. The boost optimum is much higher. What does that mean? The fitness landscape is much more narrow. That is, you give a much harder problem the second time than the first time. I want to take just a couple of minutes to illustrate to you the reason for this with an even toyer calculation. Okay? I don't know if that's a word, but anyway. So, you know, as I told you, Selection and mutation occur in different compartments in that germinal center. Mutation, replication in this dark zone, and selection in the light zone. So let's write down what mutation and replication occur in a mean field description. So n is the number of B cells sitting at position x along the breadth dimension at time, and this is replication, and this is mutation. Now here I have made mutation symmetric, has just a diffusive problem. But if I wanted to add this constant low breadth flux, that's not hard. I would add a term linear in delta n. Okay? It would not change any qualitative physics for what I want to illustrate right now. So this equation has this formal solution, where this I can call the operator D. And then a fitness landscape is imposed, Fi, at shot number I. And so that's the formal solution, and I'm going to call that the operator Fi. So at the end of the day, I start with some initial condition, and then I sequentially operate with DNF, 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 and so on, right? So now, what I want to do is that I want to maximize NF, the final state, after many shots at x equals 0. Now notice that I have used a nomenclature that may appear strange to you. The first shot is labeled L, and the last shot is labeled 1. That makes something simpler later on, okay? Algebraically. So for analytical convenience, I take the fitness landscape to be defined by a Gaussian. And you can see that this G is proportional to the integral of this. So that's like the antigen concentration. Ri is the width of this Gaussian. So that is what I want to optimize against in various shots to understand that result. Now the action of the operator D on a Gaussian or uniform distribution is trivial. So what I'd really like to do is move all these F operators to the left and operate with all the D's first. But I can't do it because these D and F operators don't commute. But there is this Baker-Campbell-Hausdorff formula which allows you to do that by taking operators like this and then making it in a simple form that they do commute, but then you do have to have this commutator. And you can cut it off at this series if the commutator is small, and that's the commutator for us. G is the antigen concentration, D is mutability. If that product is relatively small, you could do that. And you do all the arithmetic, which is actually fun. You get this sort of answer, and you find that for either Gaussian or uniform the starting points, this is what you see, where alpha is the index of the shot. And remember, the first shot is L. So the earlier the shot, the larger the value of L, alpha. So you can see earlier shots have a broader distribution at the optimum than later shots. And look at this formula. It's very nice. This diffusive form basically says, I just want to expand in the second shot only those B cells that have a chance by mutation to get to the promised land. Okay. Now let me close with two or three slides that show you that in reality the situation is much more complex than all this, but related. So, and that I'm going to describe by work that we did with Daniel Lingwood's lab and former postdoc Asaf Amitai, and the work that was aimed at eliciting these stem-specific BNAbs to influenza. Now here is a depiction of influenza. The viral spike density of influenza is the highest of all viruses we have looked at. What does that mean? That there is a steric barrier for B cells and antibodies to get into that conserved stem region. And that is part of the reason why the globular head, the variable part, is so immunodominant. And we have to account for it. Now, Daniel has many immunogens. For example, on a ferritin nanoparticle, he can put these spikes and vary the distance. He can also completely cut off the head. And we have studied, in some detail, immunization of mice with this and computational predictions and immunization with mice. I'll show you only one result. But now the computation has to be augmented. We have to account for the steric effect. So what we do is we just take the PDB structures of the immunogens and run molecular dynamics simulations, coarse-grained form, just to obtain the first passage time distribution to various sites, and thus we get the difference in the on rate of binding to different regions. And you can see here the red shows that binding to here is much higher than here. And when we do this affinity maturation calculation, we account for this steric difference as the free energy evolves by mutation. In humans, there is a particular naive or germline B cell that targets this BNAb region. And Daniel has humanized mice that exhibit similar frequency of this germline as in humans. Let me show you just one slide with comparisons. So this is our prediction of stem-specific on to off-target B cells. By on-target, I mean those that bind to the conserved region. And in blue are results when we first, every one of us has been infected by the flu. So we first study, okay, you were infected by flu, and then you gave these immunogens. And what we find is you get infected with flu, you get, this is the precursor before vaccination, that same ratio. And now you see a little bit increase when you get immunized by this. And then the prediction is, if you immunize with these two, all disappears. And the simulation gives us a reason why. There is this very strong vertical immunodominance hierarchy because of this steric business. So actually, if you just look at the headless part, the variable parts that are higher up compared to the yellow conserved region, they have many more B cells targeting after this guy, many more B cells targeting that region, and so they win the competition when you come back with these immunogens. This is the experimental result. You immunize mice, you take blood from them, and then you dilute this blood to various levels, and that's the dilution. So there's more antibodies here and more dilution here, and you measure how many antibodies bind to the stem, the entire spike. That's the black line. And then you make a mutation in this yellow region and see how many still bind. That's the red line. So the difference between them is the number of BNAbs. And you see, just like the prediction, they go away. So then is this a hopeless task? No, if you've been listening to me, it says that I have to make the variable parts of this headless immunogen different from the ones of the influenza I was infected by. And now we study this one, that's our prediction, the purple lines. It goes up. Why? After immunization with this, there are no memory B cells that are common between these two for the blue variable parts. But there are for the yellow part, and they are the ones that grow out and are expanded, which you also see in the experiments with the mice. So obviously now what we want to do is to optimize the sequence difference. And this can be done by bringing together these affinity maturation models with machine learning models of the way the sequence variation works in influenza and so on and so forth, something that we have done for COVID, and I'll be happy to discuss if someone's interested. But for now, I think my time is more than up, so I will stop. I thank you very much for listening to me on a topic that is very different from what you've heard so far.